Welcome to this explainer. Today, we're essentially going on a microscopic safari. We're going to dive into a fascinating 2017 metagenomic study by researcher Kingsley Anukam. And we're going to track exactly what happens to our own internal ecosystem when we introduce a very common, everyday medication.
But before we get into the hard data, I want you to consider this number: 2. Your gut contains about 2 kilograms of microbial biomass. To put that into perspective, that makes it this bustling, incredibly dense ecosystem of up to 100 trillion microbes. It's literally a vast, unseen rainforest living right inside you.
Now, we have to understand that this microbiome isn't just a passive collection of passengers hitching a ride. It's a highly active, kinetic metabolic factory. We're talking about essential workers that are synthesizing vitamins, producing vital enzymes, and metabolizing our food around the clock.
Okay, let's set the stage and look at the timeline. The study we're examining tracks a healthy adult male over a 90-day period. Now, he hadn't taken any antibiotics for 6 months prior. But then, he takes a standard 10-day course of a broad-spectrum antibiotic, Ampicillin, for a simple sore throat. So we're going to check in on his microbiome at day 0, day 10, and finally at day 90 to see the complete story of destruction and attempted recovery.
Part 1. A thriving gut ecosystem.
Starting our journey at day 0, the subject's gut displayed incredibly high diversity. It maintained a very precise Firmicutes-Bacteroidetes ratio of 2.4 to 1, and in the microbiology world, that specific balance is a recognized hallmark of a highly stable, healthy gut ecosystem. Everything is working in perfect harmony.
Part 2. The antibiotic impact.
But then, the ampicillin is introduced, and the drop in diversity is immediate and staggering. It acts like a biological wildfire. The total bacterial species plummeted from 401 unique species all the way down to just 98. Whole communities of foundational bacteria were simply erased in a matter of days.
The resulting chaos is honestly mind-blowing. By day 10, the environment became so disrupted that over 78% of the remaining DNA reads couldn't even be classified at the kingdom level. The ampicillin didn't just reduce the numbers, it completely broke down the fundamental architecture of the microbiome.
Part 3. Winners and Losers.
Now, in any natural disaster, there are organisms that perish and opportunistic survivors that suddenly thrive in the empty space. And the data here reveals a stark contrast. Foundational groups were nearly wiped out. Clostridium dropped from 23 species to just 2, and Streptococcus went from 19 down to 4. Meanwhile, opportunists like Villianella atypica aggressively multiplied by 1,000-fold, completely seizing the newly cleared territory.
We also have to look closely at the severe damage done specifically to the Bifidobacterium family. During that short antibiotic course, their presence shrank from 23 diverse species down to a mere 15.
So, why does that matter to us? Well, this specific loss is highly consequential for the human host. These bacteria are the heavy lifters. They break down complex carbohydrates, and they synthesize vital nutrients we absolutely need, like vitamin K, biotin, and folate. Losing Bifidobacterium basically means losing a critical piece of your own nutritional infrastructure.
Part 4. Shift in the machine.
With the core workforce decimated and new opportunists taking over, the actual metabolic output of the gut ecosystem was completely reprogrammed. The metagenomic sequencing predicted sharp, sudden functional shifts. There was a 14.14% spike in carbohydrate metabolism and a 13.64% spike in amino acid metabolism, alongside notable drops in lipid metabolism. The antibiotic fundamentally altered what the remaining microbes were actually doing.
Part 5. The slow, incomplete recovery. Let's fast forward a bit. Moving to day 90, nearly 3 months after the 10-day ampicillin course ended, the total species count rebounded to 313. Now, on the surface, that sounds promising, right? But remember, our day 0 baseline was 401, so a vast amount of biological diversity was still missing.
And mapping out that incomplete recovery reveals something really fascinating. While some of the original foundational bacteria did eventually return, entirely new species moved into the neighborhood during the recovery phase. And some of these new inhabitants were potentially drug-resistant species, permanently altering the gut's ecological makeup.
The study leaves us with this pretty striking finding from the researcher. Even after 90 days, less than half of the original species were fully recovered. The ecosystem bounced back, sure,
but it was fundamentally and perhaps permanently changed by just 10 days of medication. It really highlights how delicate our internal biological balance is. So the next time you consider taking over-the-counter antibiotics for a minor bug, you kind of have to ask yourself, is the collateral damage to your microbiome worth it? What invisible ecosystem are you permanently altering just to clear up a sore throat? Thanks so much for joining me on this explainer. Keep questioning and keep learning.